Shark antibodies are antibodies found in sharks. They are 1/10 the size of a human antibody, and this makes them more stable. The reason scientists are investigating shark antibodies is because of their capacity to cure cancer and malaria. Cancer cells are cells that don't stop multiplying. Because they are human cells, human antibodies don't recognize them as a threat and leave them. Also, as cancer multiplies, it also mutates, making it nearly impossible to cure. However, shark antibodies have been proven to target cancer, slow their growth, and even kill them. They are also able to cure most strains of malaria, unlike human antibodies. The two reasons malaria is so deadly is because 1. Human antibodies aren't able to catch the malaria parasite, and 2. The malaria parasite is constantly mutating, so there are thousands of different strains of malaria. Malaria parasites hide in red blood cells, where human antibodies can't find them. When they have multiplied enough, they destroy the red blood cell. The human antibodies will start to figure out how to bind to that strain of malaria, but before they figure out how, each parasite will have hidden in another red blood cell. While the parasite is hiding, the antibodies will still be developing to be able to bind to the parasite. When the malaria parasite breaks out of the red blood cell again, they will have changed their outer protein coat, and so the antibodies will have to figure out how to bind to them again. Shark antibodies are able to bind to the malaria parasite before they can even reach a red blood cell, and therefore stop parasites from binding to them. Even though shark antibodies are useful, we don't like sharks very much. So we take one blood sample and clone it. However, it takes a lot of time and money to sort through the different antibodies to see which one is useful and which one isn't useful. There are 6 billion different antibodies in the shark's bloodstream. However, because shark antibodies are more stable than normal antibodies, they can be made in bacterial systems, which is cheaper and faster than making any other antibody treatments.